ELISA technology is the workhorse. So it is just, let's say, sufficiently cheap to really be used routinely, and it is an established technology. The other technology with evanescence is also used in very specialized settings. So, for example, researchers, they may know BioCore. This is a plasmon surface resonance system where you can directly visualize the interaction of two molecules. What we do is, in fact, we do BioCore with fluorescence. And we do it on cheap materials. So we bring the Rolls-Royce down to the Volkswagen. So this is probably the key point. It's not that we have invented the technology, but we have made it practical.
Welcome to the Smart Biotech Scientist. I'm your host, David Brulman, and today we are diving into a revolution happening in bioprocess analytics. Ever feel like you're spending more time waiting for ELISA results than actually moving your project forward? You are not alone. In this two-part special episode, I'm joined by Bruno Oesch, a true biotech pioneer who is transforming how we approach immunoassays. As the founder of Prionics, which developed the first commercial BSC test, and now with his latest venture, Elionova, Bruno knows a thing or two about disrupting scientific workflows. Today, we'll unpack how traditional immunoassays have been both indispensable workhorses and frustrating bottlenecks in bioprocessing, and why real-time alternatives could be the game changer you have been waiting for. So grab your coffee, put those cell cultures on hold for 10 minutes, and let's make bioprocessing smarter together. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brulman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics at market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Bruno, to the Smart Biotech Scientist podcast.
Thanks, David, for inviting me. It's a great pleasure. And obviously, I'm thrilled to be able to present our technology to a wider audience.
Absolutely. It's a huge pleasure to have you on. Bruno, share something that you believe about biotech development that most people disagree with.
I guess most scientists, they're somewhat conservative. So they tend to try to increase technology incrementally. So just try and get it a little better. But in the end, it might be interesting to just take a leap, to really jump forward and get a real innovation. And this is something what I see that this is not really appreciated.
Yeah, I agree. We do live in a conservative industry, but that's why I'm excited to talk about your new innovation. But before we do that, I would like to talk about yourself, Bruno, because your career journey is fascinating. You started in prion research to founding Prionics and now your company Elionova. So draw us into your story. What inspired you to transition from academic research to entrepreneurship and now starting several companies?
Well, this is difficult to say because originally I was really on the track to get an academic career. So I was really working towards an academic career. I was working on prion already as a PhD student. Then went as a postdoc to the US to Stan Prusiner, who later on got the Nobel Prize with some of my data. And later on, I went back to Zurich and had my own research group. And then the BSE came along, mad cow disease. And we, in fact, had thought in the lab, we should just do a test. And then we thought, okay, let's try and figure out who might be interested. We went to the veterinary authorities. We went to industry like Roche, Novartis, and so on. Tried to figure out whether somebody would be interested, and nobody was interested. And then in the end, we said, okay, we have to do it on our own. There is nobody who really would like to push this. And in the end, we in fact found out that the veterinary authorities had been asked also by the industry whether such a test would be needed. And they in fact then said, no, it's not needed. But ignorant as we were, we went on and finished our BSE test, and that was it. That's what's the start. And then since we didn't find anybody to commercialize it, we had to found our own company, and this was Prionics. So after that, I thought it was always fascinating to bring science into the real world, namely to do something practical with it. The academic results are also fascinating. For me, it's fascinating to solve a problem, but I find it even more fascinating if the problem also has some real application or the solution has real application.
Now, once again, you're bringing science into the real world, and you are disrupting the analytical space. But let's start with the basics, because ELISA assays have been the workhorses in bioprocessing for many decades to do quality control for ensuring product safety and efficacy. Could you walk us through how ELISA actually works and why this technique has remained so fundamental in various areas of bioprocessing and even larger space in diagnostic and the whole biotech world?
ELISAs have been developed in the 1970s, and this was really a breakthrough for analyzing proteins. So the technology works such that you, for example, a catching antibody at the bottom of a plastic plate. Then you add your analyte, let it bind, wash away the rest of the analyte. Then you bind the detector antibody, wash away the rest of the detector antibody, and then finally you come with a detecting antibody which has an enzyme coupled to it, a secondary antibody. And in the end, you have, let's say, a color development or fluorescence or whatever. And then you can see the result. Then you can quantify the results. How much signal do you have? So this is a process which involves a lot of washing, incubation, and so on. And most researchers, they have their own ELISA, so this usually takes 4 hours or more overnight, whatever. So it takes quite a while to get the result. This is the classical technology, and it has been surviving because it is so useful. So it is really the workhorse of biochemical laboratories. So our technology, going now to the ELIODX, we call it. So it's the evanescence technology. This is now a major step forward, and I have to admit that we haven't been the first ones to use this. So there have been other companies, like there was a company in Basel, Septosense. They were exquisitely sensitive, but then they got sold. And one of their problems was that their technology was fantastic, but was too expensive. So just to do an experiment, if the basic cost is 50 francs or more for one experiment or for one data point, then this is not something you can or you would routinely do. And as in an ELISA, where, the plate costs at best, let's say 1-2 francs. If you buy the ELISA, it's maybe 5 to 10 francs per well, something like that. So that is something where the technology existed, but it was way too expensive. And in addition, it required glass substrate. So it required optically clean substrate. So maybe I quickly should go into how the technology works. So what we do...
Maybe just before we do that, I have another follow-up question just to understand, because, it definitely makes sense to have a better technology, but I'm just wondering, why is ELISA still around and actually used that often? Is it because it's cheap, or are there some other advantages? Because why has nobody else tried to commercialize or develop a better technology?
ELISA technology is the workhorse. So it is just, let's say, sufficiently cheap to really be used routinely, and it is an established technology. The other technology with evanescence is also used in very specialized, let's say, settings. So, for example, researchers, they may know BioCore. This is a plasmon surface resonance system where you can directly visualize the interaction of two molecules. What we do is, in fact, we do BioCore with fluorescence. And we do it on cheap materials. So we bring the Rolls-Royce down to the Volkswagen. So this is probably the key point. It's not that we have invented the technology, but we have made it practical.
Okay, I see. Yeah, you made it accessible
Yes.
to a wider variety of people. Okay, I see. So how does the technology work in detail?
So usually you have to incubate and wash in order to get rid of the excess analyte or excess detection antibody, because you always look at the whole well, at the whole cavity. So what we do is we only look at the very, very bottom of the cavity where the reaction, so the capture reaction, for example, where the capture antibody binds to the antigen, and the detection antibody then binds also to the antigen. So we form a complex at the bottom of the well. In order to see it in a regular ELISA, you have to wash away the other molecules. What we do is we come in with a laser at the bottom of the cavity. The laser is totally reflected, and this creates a physical phenomenon which is called evanescence field. So this is a field of 200 nanometers of laser light in the liquid at the very bottom of the well. So now we only see the fluorescence molecules which are at the very bottom of the well. So when we start the measurement at time zero, obviously there is just, let's say, the random molecule which flows into this field and gets out again. So if we have no analyte, this is all we see, this random movement in and out. So this gives a horizontal line. And this is what we say, okay, this is our background. Now, as the reaction progresses, more and more of the complexes form. So the capture antibody binds the analyte, and the analyte binds the detection antibody with a fluorescent molecule. So more and more fluorescent molecules are attached to the bottom of the well, so more and more fluorescence is being seen. And since we observe the reaction over time, so we measure the same well about every 30 seconds. So this means we then have a time course of the reaction. And we do not look at the height of the signal, but we look at the increase of the signal over time. So we take the slope. And the slope is proportional to the concentration. So if we are at the bottom, it's flat with no analyte, a little analyte, more analyte, more analyte, more analyte. And we take the slope and plot it against the concentration for a reference curve, and that's it. And then we have a reference curve, and now we can, from an unknown sample, we can measure the slope and calculate back the concentration. And the trick is we do this in 10 minutes. So we really look at the immune reaction in real time. One additional advantage we have is sometimes you have background. In a static system, what you see is, if you have a high background, you see this as a signal. For us, it means that instead of starting at the very bottom, we start a little higher. But then we just see the increase is the same. So we just start from a higher level. So we have automatic background subtraction because we only look at the slope and not at the absolute level of the signal. Slope, so signal over time, increase of signal over time. This is our key.
And to what extent can you measure, for instance, a cell culture broth? Do you have to wash that? Because obviously you need to be able to measure it, or you don't have any matrix effects or so on with your technology?
Well, certainly we also will have some matrix effect. Then you have to have a reference curve in the same matrix. This is as simple as that.
Yeah. And you probably need to do some dilution. If it's very concentrated, you probably need to dilute first the sample to be in a meaningful range, I imagine.
Oh, yeah. So this is the same in an ELISA. You need to know the range where you can measure your sample. And if you measure outside that range, if it's too low, then you just have no signal. If it's too high, what we get would then be something like called a high dose hook effect. So this would mean that we in fact bind some analyte at the bottom, and we also bind analyte in the solution with the fluorescent antibody, but they're not forming the complex at the bottom because there's too much analyte. So in the end, what we see is, in fact, we see the signal increase with concentration, but at a certain point, then it decreases again. So we need to know roughly what concentration. And in that sense, for example, we have now for CRP, C-reactive protein in plasma and serum. There you can have very low amounts, which are normal, or medium amounts, high amounts, very high amount. And what we do then is we just test 3 different dilutions, 10-fold, 100-fold, 1,000-fold dilution, just on the same cuvette. So the cuvette is one of our devices. So these are the cavities.
How big are the cavities? What is the volume of a cavity?
So we use 20 microliter of sample.
Okay.
So what we do then is we just do, for example, a negative control, 10-fold, 100-fold, 1,000-fold dilution in duplicate. We have one cuvette, and we then insert this into the machine. So we pipette this into the machine, and 10 minutes later, we have the results. So what we then see, for example, if we have very high concentrations, the lowest dilution will have, for example, less signal than the 100-fold or the 1,000-fold dilution. And then we know exactly we are in the high dose hook effect.
And how many samples can you do at the same time? Just a number, or could you even do 96 well plates at the moment in your device?
This is something which a lot of people ask. Unfortunately, technically, we're not there yet.
Yeah.
So at the moment, we just do one cuvette at a time.
Okay, I see.
So for getting quick results, that is definitely the right thing. If you have thousands of samples, then I would still recommend, at least at the moment, that you still use ELISA. The other thing is, if you have so many samples, usually it's not important whether you have the result in 3 hours or 2 days, because it's a huge experiment and you just want, let's say, the results all together at a certain time point. However, let's assume you have done some affinity chromatography, you get some elution, you want to know where is your protein. If you then have to do an ELISA, it means you have to do all the incubation and you probably get the result the next day. With us, we can just do, let's say, 3, 4, 5 cuvettes, and within an hour, you get the result and you can continue working.
And that's exactly where I see a huge advantage of this technology, because in bioprocessing, a lot of people are moving towards more continuous processing where you want to have almost real-time results. So this could be definitely a game changer. You mentioned in downstream, you want to measure your residual host cell proteins, or after your capture step or another chromatography step, you could easily do that and see, oh yeah, we are right, we're wrong.
Right now, we have a project with a company. They're doing fermentation and they want to look at the production. And by the way, what they didn't know, what we then found, that at a certain point after the maximum growth has arrived, so that the product, the way they want it, is in fact not at the maximum growth, but sometime later. So now they can in fact measure very quickly at what time point should we harvest. And this is something where you get the result back in 10 minutes. And this is just in time for reacting. Otherwise, you would not have the possibility to measure this. You can take samples, but in the end, you have to harvest at some point when you do not know the result. And in that sense, you may have harvested the wrong time. While with our technology, you could know exactly the time point, or you could take the decision maybe 10 minutes too late, but if 10 minutes really are important, we could even shorten the the measurement time.
Yeah, and even for companies that are using now predictive modeling, as you have well characterized your process, as you're then taking these samples, okay, my process in this range, and according to our model, we're going to harvest tomorrow. So you can plan for that, or you can tune certain process parameter in order to get the right titer or a product quality. So I see a lot of benefit of such a real-time method for, I'd say, a variety of different use cases. I have just one other question about the assay itself, because you highlighted, okay, one of the advantages is the time, it's 10 minutes. But what about the precision and the accuracy? How does this compare to other assays? You mentioned BioCore or traditional ELISA.
In terms of sensitivity, with regard to traditional ELISA, we are equally sensitive or better. Mm-hmm. So in that sense, we can easily replace an ELISA. With regard to BioCore, BioCore has no label. So BioCore, in fact, is not very sensitive. It is a useful technology to look at the affinity of an antibody to its antigen and so on, but it is not a method which you would use on a routine basis. And in addition, it's not very sensitive. I guess it's 100-fold less sensitive or something like that. So this was the other company I mentioned, Septosense. They were exquisitely sensitive. So they were, in fact, more sensitive than we are, but they had the problem of the costly basic materials. So that is the trade-off. Unfortunately, do not get everything in the whatever you would like to have. So we now have disposables, we have the speed, we have the sensitivity, at least ELISA type sensitivity. And I think that this is something which is established also in laboratories, that this sensitivity usually is fine. In the end, sensitivity always depends on the antibody that you use. So in order to become more sensitive, usually you have to get really very good antibodies, high affinity antibodies, and so on. If we have high affinity antibodies, we increase our sensitivity as well.
And how does your assay compare cost-wise? Because obviously you have less work because you have no washing. It's easy to handle. Is that similar to an ELISA? Is it cheaper? Is it more expensive? How is that?
So material-wise, since we use the same, so this here, this cuvette, this is ELISA grade polystyrene. So it's the very same material that you have in the ELISA plate. In fact, we even irradiate also the plastic in order to make it binding. So this is what is done with ELISA plates. High binding ELISA plates, they're irradiated, and then the surface is somehow activated and binds more proteins. We do the same thing. So this is exactly the same. So usually to bring an ELISA to our system, you do not have to change anything. You can use the same condition for coating. You can use the same condition for incubation. It's the very same. And I say we can bring an ELISA within 10 days onto our system. And we've done it more than once.
That's 10 days to develop the whole method. So if you have a method running, for instance, for one protein in 10 days, it works.
Yeah, if you have the materials for an ELISA, I say in 10 days you have an ELIODX assay.
What are, I don't know if you have already had these discussions with potential customers, for those who work in a regulated space, pharmaceutical producer, you said one of your client is in the fermentation space. What kind of regulatory or validation challenges do you foresee, or that would be really the same as with the current assays we have in the industry already?
The exact same regulation. So if there is, let's say, GLP, GMP requirements, we would have to comply with that. And if it's in vitro diagnostics, we have to comply with that. So in the end, it's the very same thing. It also means that, for example, for IVD, it will take us a while because IVD has really the highest regulatory hurdles. And there is also a bottleneck in the approval of diagnostic tests because with the new IVDR regulation in Europe, all the tests have to be recertified. And there are only 5 notified bodies for all of Europe. So you can imagine they queuing outside the door. And I've talked to a group and they said, yes, Bruno, it's actually one of my former employees at Prionics. Bruno, you're very welcome. But I have to tell you, if you come, we mostly work for Abbott. And if Abbott has an urgent problem, they go in front of the line. And that's it. That's the way they work. That's, as a small player, you do not have any chance in that sense. You cannot force anything. And obviously the big players, they have much more influence. And this is as in real life. I mean, look at the politics.
That wraps up part 1 of our fascinating conversation with Bruno Oesch, who is the CEO and the founder of Elionova, on reimagining immunoassays for the modern bioprocess landscape. We've explored why ELISAs have remained fundamental despite their limitations and how real-time technologies are poised to transform your workflows. Join us in part 2, where Bruno will dive deeper into the revolutionary EVA technology behind ElioDX and share his insights as a serial biotech entrepreneur. Before you go, are you struggling to choose the right technology for your bioprocess development? I have helped many biotech companies navigate these decisions to accelerate their path to market. If you'd like personalized guidance that fits your unique process challenges, book a free consultation with me at brumanconsulting.com, and I'll see you in part 2. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.